Good morning, students. Hope you'll be fine at your respective places. In continuation to the last topic, what we have started the seed. We have given the little bit of introduction, and in that introduction, we have already discussed in the second video that various types of the seeds are there, and that basis we have made it on the basis of the number of cotyledons and on the presence or absence of the reserve food, that is the endosperm. And we have discussed in the dicots, we have got two types of seeds. We have got one endospermic and another one is called a non-endospermic. And non-endospermic seed we have taken. Today we are going to discuss another type, that is the endospermic dicot seed. इसका स्टार्ट हम पढ़ लेंगे. That is the endospermic dicot seed. That means both endosperm, or we can call it as the albuminous dicot seed. We know that when we draw the structure of this seed, what we will find that it is somewhat broader seed. Yeah, we are giving you the example जो हमने वहां पर दिया था, that is the castor seed के हमने वहां पर example दिया. If we draw the diagram, we will find the diagram like this. If we draw the diagram will be like this. Like this. Somewhat broader as compared to that of the non-endospermic dicot seed. Here we will see first जो इसमें आ जाएगा, जो outer covering इसकी है, that is the seed coat. Seed coat यहाँ पर what we will see, the other two. First one is the जो outermost, that is the testa, and the inner one that is called as the tegmen. लेकिन in this case testa and tegmen, they are there, but they are fused. Now how we will see that there are two layers, that there is a testa, there is the tegmen seeds. How we can see that? If we see testa की जो बाहर वाला इसका है, that is the outer coat, it is somewhat patchy. And if we go on the inner side, that is very much smooth. This is the outer, this is the inner. Outer जो हमारे पास है, it is somewhat patchy, it is somewhat designed, while as on the inner side it is soft. But when these two, they are somewhat fused together, we cannot separate them, but it is only visible that जो इसका outer side इसका seed coat का है, that is patchy, and that patchy or that designed side जो है, that is the testa, and the inner one जो है, that is smooth like that of the typical dicot seed, that is the soft, and that is this tegmen. So what we will see here, that this is the seed coat. Now on the micropylar end, like this, we will find here these two, what we will call as the outgrowths or protrusions are found there. And what are these? It is called as the caruncle. These outgrowths are called as the caruncle, and this caruncle is considered as the third integument. This is considered as the third integument. In a typical, अगर हम बात कर लेंगे dicot, in a typical dicot seed की अगर हम बात कर लेंगे, जो वहां पर हम ovule देखते हैं, what we will see it like this. We will find the structure like this. This is the one integument. This is the second integument. Then there is the third integument which is found there. This third integument is found there, and this third integument, when this ovule it has developed into the seed, this this is the testa, it gets converted into testa, this gets converted into the tegmen, and this third it gets converted into this, it develops into this, that is this outgrowths which are present at the micropylar end. This is called as the carunc. Now inside what we will see in this seed, like that of this endosperm, what we will see, there is this embryonic axis, like this. There is this embryonic axis, like this. I will show you like this. If this is the embryonic, this is the embryonic axis, and these are these two my hands, these are the what we call as the cotyledons, because we are going to study the dicot seed. These are the two. If I will see there, you will find them as elongated. When I turn my hand like this, you can see only one cotyledon, but that is somewhat flatted cotyledon. लेकिन जो हमारे पास this embryonic axis है, that is not visible to this. But in this diagram, I will try to show you this figure like this embryonic axis, as well as these narrow, these longish, what we call as this cotyledons also. If I draw the diagram, this is simply the where they meet. This is the cotyledonal, this part. This is the cotyledonal node. Now this part which is present towards this chalazal end, this is gives rise to the plumule, and which is found towards the micropylar end, it gives rise to the radicle. This plumule, it gives rise to the stem, and this radicle, it gives rise to the root. In between these two, what we will see, cotyledon node and this plumule, there is a similar structure what we have studied, that is the epicotyle. And in between these two, what we will see, I will find another structure between this cotyledon node and this radicle, that is called as the hypocotyle. Now this portion, these two flat, these are the cotyledons. These are the cotyledons. These two, these are the cotyledons. Now, since we are going to discuss this endospermic, whatever the space that is left after this cotyledons, whatever the space that is left, this space, this जहाँ पर मैंने ये dotted ये क्रॉस डाला, this is the endosperm. This is the endosperm. Whatever the space that is left, this is the cotyledons. So what we will see here, this endosperm, it is cotyledon, this part. So this portion, that is the endosperm. अब जो यहाँ पर जो रिजर्व फूड, जो फूड यहाँ पर स्टोर है, इन केस ऑफ दिस एंडोस्पर्म, दैट इज फैटी. That is fatty. Now, how do you come to know that is the fatty? When this castor seed is crushed, we get the oil, and that oil is fatty in nature. So the food, reserve food यहाँ पर जो endosperm है, it is fatty in nature. So what we have studied, that in case of this endospermic dicot seed, यहाँ पर जो seed coat है, There are the two seed को there are the two layers, that is the test and tegmen, but they are fused. अब हमको फ्यूज कैसे पता चलता है? जो बाहर वाला इसका हिस्सा है, it is somewhat patchy, it is somewhat designed, and the inner is the typical dicot coat, that is this soft, and there is the embryonic node. Again, there is the one that makes it different from this non-endospermic, that on the micropylar end there are the two outgrowths which are called as a caruncle, and that caruncle is the considered as the third integument, while as in the typical ovule we have only got the two integuments. Then there are the cotyledon which are somewhat narrower. When we like this, when we put them this on the side way, that it becomes somewhat flat, and there is this what we call as the rest of the space यहाँ पर है, that is this endosperm, and that endosperm is fatty. वो हमको कैसे पता चल जाता है? When we crush that seed, we get the oil, and that oil is mainly the fatty. This was all about this dicot endospermic seed, and tomorrow we are going to discuss the monocot seeds. Until then, goodbye. God bless you. Thanks.